Hi class and welcome to lab 7. This lab will be on membrane transport processes and we're going to be looking at osmosis. The lab worksheet, I'm going to go with you right now and we'll kind of go through the activities that we would be performing if we were in person. We did go over osmosis and tonicity a little bit in one of the lecture chapters, so you can go back and refer to that for a little bit more help. But basically, the passage of molecules into and out of the cell is regulated by the plasma membrane. The membrane is said to be selectively permeable because it only allows certain molecules to cross freely. Some of these molecules may simply diffuse across the plasma membrane, but large substances may need to be transported by carrier proteins. Diffusion is what we call the net movement of molecules from a region of high to low concentration. And osmosis, which is what we're going to focus on today, is specifically the diffusion of water across a differential permeable membrane from an area of higher water concentration to an area of low water concentration. A solution always contains a solute, the particles being dissolved, and the solvent, or the liquid that dissolves the solute. And the tonicity is the relative concentration of solute and solvent outside of a cell compared to inside of the cell. So for the first activity, we take you through a series of pictures based on the experiment and what the experiment would show if we were in person. We are going to be using what we call dialysis tubing. It looks solid, but it actually contains pores of a certain size. And the dialysis tubing is going to mimic the selectively permeable nature of the membrane. So that just means that the dialysis tubing will only allow certain substances to pass through. Also, we're going to be using the reagent IKI, which is iodine, and iodine or IKI tests for the presence of starch. So just remember that IKI is the reagent that tests for the presence of starch. It's normally amber or yellow in color. And when it comes into contact with starch, it will turn dark blue or black. So keep that in mind, and you'll see what I mean when we go through the experiment. So we're going to take a look at the experiment and note the color of each and then answer the questions. So activity 1 is diffusion through this selectively permeable membrane, and we're going to use dialysis tubing as our selectively permeable membrane. We would use clips, we need a beaker, we need starch as the substance we're going to test for to see what moves. And then this IKI bottle again stands for the reagent that tests for the presence of starch. So activity 1, to set up this experiment, we would put some starch into the dialysis tubing bag. You can see that the starch normal has kind of a cloudy white color. And then we would set up our beaker with DI water and IKI, that reagent. So you can see here, the IKI reagent is normally like an amber or yellow color. But if IKI comes into presence with starch, it will turn very dark blue. So just keep that in mind, dark blue or almost black. So this is our experiment setup. So this is what we're going to do. What we'll do is we'll put the dialysis tubing with a starch. So you can see it has that white cloudy color. We're going to place it into the beaker that contains the IKI reagent as well as the DI water. And then we're going to wait several minutes, whether it's a half an hour, to see if any starch either leaves the bag or if IKI, the reagent, goes into the bag. And we'll know that because the color will change when it comes into presence with starch. So remember, IKI will change to a dark blue or black when it comes into the contact with starch. So we're going to see what happens after a while to see if any molecules are getting through this dialysis tubing, which again is the selectively permeable membrane. So here's some questions regarding setup that you can answer on your own. And I think we covered most of them. It just asks you if you understood how we set up the experiment. And then after 30 minutes, let's see what happens. So you can already see a color change. So let's take the bag with the starch out. You can see here that the bag has turned either kind of a dark blue or black color. And what that means, I'll let you guys figure it out. Starch was inside the bag, and we put the bag inside of the beaker with IKI, that reagent. And what that means is basically the IKI reagent was able to pass through the dialysis tubing. So its molecules were small enough to pass through. And when that happened, it changed the contents of the inside of the bag dark blue because it came into presence with starch. So we know that IKI, this iodine reagent, was able to get into the bag because the bag went from a cloudy color to a white color. The beaker, however, still looks the same. So my question for you is, did any starch leave the bag and get into the beaker? And the answer is no, because the beaker still has that natural yellow amber color because IKI was put in it, and it would only turn the dark blue if it came into contact with starch. So we can see here that the starch remained in the dialysis tubing and did not leave it at all. So we know that starch, its molecules were too large to pass through the dialysis tubing bag. But the IKI reagent was small enough to get through the pores because you can see that it came into contact with the starch and to change the color. I hope that made sense a little bit. There's some more questions to ask at the end. And then the next activity takes you to a website where you'll be watching 3 different experiment videos and then recording your observations in these columns and then answering the questions on the next page. So this video, you should be able to open this link with Safari. I always like to try Google Chrome because that might work better. But here's the Safari link with that, with those experiments. All right, so you'll watch those videos with the experiments, record your observations, answer a couple of questions, analysis and conclusion questions. And then the last activity takes you through an examination of plasmolysis using Elodia. And this will take you through a plant cell and how the plant cell will change when placed into a salt solution. For this part of the lab, you can pretty much read through the slides and should take you through how to answer all the questions, what happens to the cell contents in a hypotonic environment, what happens to the cells in a hypertonic environment. And remember the words hypotonic and hypertonic. So a hypotonic environment refers to something, the cell is going into a solution that's less concentrated, that's hypotonic. So these are the plant cells in distilled water. And these are the plant cells in a hypertonic environment, putting it into a solution which is more highly concentrated. So these are the materials that were being used for this experiment. A wet mount slide of the plant cell, 4% sodium chloride, just salt that will be added later. And this will show the wet mount slide of Elodia, the plant cell, before adding the salt concentration. And then you'll be able to see underneath the microscope. So this will be just a hypotonic solution that you're putting the plant cell in. And then this will be the hypertonic solution that the plant cells were in. So you're adding salt water to the slide to see how the plant cells change. So what they do when placed in a hypertonic and hypotonic solution. And this is just an examination, labeling some of the pictures there to help you with your observations. And then you can answer the questions. And that takes you through this lab 7 that's due on Sunday. Again, like always, you'll have a quiz on this the following week. And if you have any questions, please always remember to shoot me a message on Canvas or an email. And I hope you guys are all doing well.